Take my podium.
It's picking up to here. Okay, I'll go ahead and get started because I'm not going to get us.
Mary's getting started, everybody.
I don't want to. I don't want to take the whole break up. So, I'm Mary. I'm the geneticist with the Forest Service ash breeding program. So I'm here representing quite a few other people. Dave's been chiming in. I'm not sure if Jennifer's online listening to. And then we have quite a lot of. We have a number of our team who are embedded from Holden Forest and Garden. And that's our current list, along with alumni and student interns. So, this is, roughly 20 years, a little bit over 20 years worth of work kind of summarized. So lots of people have contributed. So just kind of taking a step back and thinking about what we've been talking about, but maybe with a slightly different lens. We're like incredibly lucky, I think, in North America, because so many of our tree species have just tremendous diversity. Genetic diversity. And there's a lot of things that kind of feed into why we have the genetic resources we have in our trees. We have just like monstrous ranges, like half continental ranges, not for all the species, but for some. And that gives us huge numbers of individuals. I mean, thousands upon thousands of trees out there on the landscape. So we have wind pollination systems, which, and we have sex systems, and self-incompatibility systems, which require outcrossing. So we get a lot of heterozygosity, which is 2 alleles at each locus. That's a way to help retain more different alleles in a population. There's population substructuring. We know we have continental clines from north to south, and we have a drought cline or a moisture cline from west to east that we see in a lot of different species when we look at the geographic variation. And then we have a lot of local adaptation, whether it's adaptation to a swamp or to a stream, being in a stream corridor, or just our local elevation adaptations. That's what's out west. Elevation is a lot of our local adaptation. And we have some of that like here in this region of the country too. Littler mountains, but they're still there. And then there's the genetic processes. When you tend to take genetics as an undergrad, you talked about things like founder effect and drift and some others that we've all forgotten. But you roll all those things together. And that's all what's happening out on the landscape. And we have many, it's a, embarrassment of riches in our genetic diversity, I think, in our trees. And for a lot of our species, and all of the species of ashes we've been discussing kind of fall into this, of our North American ash, fall into this system and have this diversity. So what that results in is that you have a lot of retention of rare alleles. And so when something happens, sometimes a rare allele can be helpful. Sometimes when something happens, a rare allele can be helpful. So this is one of my favorite of the early "Don't Move Firewood" cartoons from like somewhere in Ohio. I think this ran in I think my dad sent this to me from the Springfield, Ohio paper. So anyway, these rare alleles can come under selection pressure with this new pest, and we think that's what's happening in ash. And it's certainly something that the more we look carefully, we're seeing in some other species too. So that's the perspective from which our group kind of approaches the problem. I mean, the threat's not I think it can't be understated. We've talked about this quite a bit. So I'm just gonna move forward. So our approach is to use the tools and the processes of plant breeding to try to address this ash EAB problem. And so just to kind of go over like, how do you have a breed? What are the steps in a breeding program? And I've kind of, I put these little headers in, and I'm just testing them out in my talk again. So I'm kind of liking them though. Good trees. We want to go out and find good trees on the landscape. That's selection. That's the far left. And as we move to the right, we're going to show that they're good trees. It's an actual reproducible phenotype. We can do that both in a potted test, like you see in the middle. And if you go down, you see a field test cartoon where you plant the trees in the field. And then at the top, you see a representation of a cross. There's one tree crossed another tree, and there's all their kids. So we're going to put those down into the potted assay and down into the field. So we're going to test both the selections that we're going to use as parents, and we're going to test them and the progeny both in the bioassays and in the field. We're going to roll all that data into one big data file and use it to choose our best trees. We're going to put those into a seed orchard, which we're going to manage for seed production, good crossing, good tree health, so we get lots of good healthy seed that'll germinate. That's our good seed. And then, we add our nursery pipe and our restoration practices, and we can have good forests. So that's our hope. That's our hope with this. That's our scheme, what we're trying to do. So we've been talking about lingering ash. They were originally, this is good trees, our first step in good trees. We've originally found them singly, and as we've looked, we've found a couple clusters. And one of the clusters, there's a few pictures of the cluster on the right there, so I'll walk you through those. The upper right picture, so this is Alum Creek. It's a flood control impoundment near our lab, and we went out and actually Dave was hiking with his family and saw a tree, so we went back to check it out. On the way out, you walk through that area in the upper right, which we like nicknamed the boneyard. All those dead trees on the ground are ash. And so it's just, there's not anything there alive. But you keep going down the trail and you come up to trees like the two pictured below. Those are decent sized, live, healthy crowned ash trees. And they're, a quarter mile from the boneyard. So something's going on. And when we got in there, we'd find one, and then from that tree, we could see another. And from that tree, we could see another. So this is definitely a cluster. We think based on tree staff, we may have another third cluster to go check out in Ohio. So we're starting to see clusters too. We're not sure what that means or how those originate or what's going on with those. So that'll be a talk for a couple of years when we sort that out. But they're out there, and they have the lingering ash fall into a range of phenotypes, exactly what you would expect from most sort of response to a pest system. Almost all the trees are going to be like the one on the left, susceptible, dead crown, dead bowl. The middle and the right are trees, examples. These are winter pictures, but they're trees that have healthy crowns in the summer, but some of them have a lot of damage. And so we've talked about selection criteria, size and whatnot, but what I wanted to emphasize was this is not immunity. When we're talking about a lingering ash in phenotype, we're not talking about a tree that looked like trees did before, where we've got no fencing and very little weed control, and we're going to see what happens. So spoiler alert, deer browse happens. So, but anyway, we're going to need more trees. So we're asking folks, especially in our region, in our appropriate region for our first orchard, the tree snap trees, and we're always looking for planting sites. We've kind of talked about that already. But I will point out too that not only can you tree snap ash, there's lots of other fun trees that people are looking for distribution and health data on too, including sassafras and butternut. Let's give them some love. And these are just a couple teasers. We do look a little bit at blue ash. Can't obviously get into this today, but this guy with the heart is our one lingering blue ash compared to a number of other blue ash. And so 2 points here. One is there's some lingering blue ash. And the second is when you actually put the eggs on blue ash, a lot of them do terrible. So yeah, there's more to sort out on blue ash. We're also looking at black ash. The black ash has got, I don't have time to really get into it, but black ash has got kind of an overreaction to the EAB attack. So there's a second kind of level of being able to detect the larvae there and kill it, but stop killing before you kill your whole self. So there are some lingering black ash that kind of do that, but we think they may be rarer because you're kind of selecting for a double phenotype, not just one response. Anyway, and we're looking at seedlings in blue and black ash too. And what's interesting about blue and black ash seedlings, these are unselected seed lots, pre-EAB, nothing. And there is more resistance. The two far right bars are green ash. You've got blue in the middle and black on the left. There is a lot more, and again, the orange portion of this bar is the host killed from the bioassay. So there's a lot more host killing in unselected blue and black ash than there is in green. So you asked what you can do in Texas. Might throw some open pollinated pre-EAB trees out in the field from like albicans and see, get them in place before EAB comes through and see if you get lucky. There's some possibilities there too. But coming back to our focus and then putting it into the context for today, we're driving towards our orchards. We want good seeds and good forests. So to get there, we're getting good to have good parent phenotypes. We have to have a heritable trait. We have to manage our seed orchard for seed production. The seed has to be competent to germinate. It has to grow in the nursery. You have to be able to take it out to the field and have it live, both initially and be competitive as it grows in a forest. If you don't get all of that, you're not going to get ash back on the landscape. So this is sort of our more detailed workflow. Every one of these boxes is an experiment or an operational process that we do as part of the ash breeding program every year. So we sat down as a group and talked about all the things we do and which one of those were impacted by either having the wrong ID on a tree or having a mixed population of green and white ash. And I color coded the steps we thought might be impacted. So, yeah, so many of them. So talking more specifically about a few, if we think about selecting trees, I mean, we can go out and look at trees. We can assess the tree crown class. We can look at the overall health of the tree, the size of the tree. We can do all that. We don't even know what kind of ash it is. We can do that for anybody. It's when we start to look at the relative health of the trees or try to collect a certain number of trees of a certain species for our collection that we run into problems with the IDs. We know, especially in our case, we focused mostly on green and white ash. So we know there's a difference in a mixed stand and how quickly they're attacked. So if we go in at a certain time point, we may be selecting white ash as healthier or biasing against green ash just because of that preference difference. And if we are able to, you know, confidently identify which species it is, we can kind of correct for that. But if we can't identify, then we're probably maybe biasing our collection. And there's, based on our numbers on our inventory, I'd say we did some of that as part of our process. So, so we're having to switch. We were focusing on getting more white. Now we're thinking we need to really look harder for green ash in Ohio because we don't have as much green ash as we thought we had. The parent generation, the trees actually select the lingering ash. When we phenotype those, we do that big bioassay. We can score in the field and we can combine that data just fine without, you know, if it's say it's a mixed population, that all works. But when we try to rank the trees and select the top trees, we would expect our white ash, if it's a mixed population, our white ash to maybe rank higher. Maybe not. As we get into more resistant trees, that bias may disappear. So we have to see how this works out in actual work. But we're concerned that we're biasing there. And then the other thing that's kind of hard is when you get ready to put one like that clonal test at Holden. I talked about we're starting to get EAB damage. We're going to have mortality. We have to pick a year to rank these trees and go who's the best and who's not. If we don't know, if our controls are white ash and most of our trees are green, we're probably not at the right endpoint for when we're going to rank the trees. We're probably too early if our controls are green, and we're too late for the green ash if our control is white. So picking the right control and picking the right moment, it's the ranking that gets kind of sideways. Similarly, when we start to phenotype the progeny generation, there's a lot of things that we can do. We can do like all the things we did with the parents. In addition, we can let those stumps resprout and plant them. That's no problem. White and green are kind of the same for that. And selecting within a family shouldn't be impacted because all those guys have the same, presumably within a species, if it's a hybrid family, they're all the same hybrid, or more likely it's a pure species family, and we can select within the family without a problem. It's the comparing. So when you go to compare the families, if you've got some families that are white and some that are green, we might be getting some bias in our selection. So it's relative rankings again, looking for the overall top performers. The same design problems with the progeny field test as we have with the clonal field test, as far as finding the right controls and then choosing that moment when we're going to actually score and say who's best and who's worst. And we've talked some about the field, the field preference that was seen. I will tell you that we planted, we have a planting in Worcester, and we put in a whole bunch of couple seedling blocks and a grafted block. And we were concerned at the time about, EAB populations were crashing in Ohio. And we're like, we want to make sure we keep EAB around. So we put a big border row all the way around, and I alternated white and green ash. And these were just from a nursery, conservation grade. And in that planting, we did see all of the pen attack before the white. So what we've seen in the woods, we did see in our breeding program planting also. So it's not, I think that's, these biases, potential biases, or this preference is going to be, at least in susceptible material, is definitely going to be part of our plantings too. So we are going to have to deal with these issues. The big problem, so we talked a little bit about, we're talking about the problems and where we think we may have problems. So this is where, and this is again, something that we've gotten a lot of help from Zane and other folks in Meg's lab with, is we've got some big problems in compatibility and reproduction. So these examples for the next couple slides, there's some numbers from the breeding program. But I think what I really worry about is every one of these problems. If we have any control crosses to make control crosses for our breeding program, it's going to be the same problems translated through to breeding orchards. And in breeding orchards, it's going to kill us. So we can work around things in the breeding program. That's kind of, it's research. We work around problems all the time, but we're not going to have productive orchards if we don't do the right things. So the first step where we see a drop off, where we believe we're seeing a drop off based on species matching, is in going from pollinating much flowers and how many seed do you get. So the table here, the parenthesis is the number of crosses that were attempted, and then the number is the percentage of flowers that went on to produce a seed that we collected. And it's set up as a grid. You see, so you can see upper left is white by white and bottom right's pen by pen. And then so we had all these trees. We had called them one thing or another. Dave made the crosses from mostly Dave. And then we did the GBS sequencing of the trees, and we figured out that some of them were miscoded, and we were actually doing white by green crosses. So we didn't necessarily intentionally try to do this many hybrid crosses, but that's what we think is going on. And you can see that, like, especially the pen female and the white male group, I mean, the seed set's very, very low, and that's a fair number of crosses. So it's a lot of different combinations. And we know this isn't the mating types. And some of this is the seed is initially set and then kind of comes off the tree midsummer. I don't know if you've been hanging around, if you've watched ash trees really carefully, they do kind of in Ohio, like late July, early August, they'll drop a lot of seed partway through the maturation. And we see a lot of loss at that stage in some of these cross species crosses. So, you know, I don't think we can tolerate this kind of seed set in a seed orchard if we're in the red boxes. So this is the next step, germinating the trees and actually getting a seedling that we're growing in the nursery. So this table's set up, that's percent that germinated initially, the number of trees that is in the germ column, and the number that actually made it from germinate to like a little tree in a pot is in the trees column. And this is a little muddier. You know, we don't have a bad germination percentage in that green by white box on the bottom left. But you can see then we get a big drop off in the seed from germinate to seedling. And the same in the upper right. We just get a lot. I mean, that's a 50% loss at that step. You know, a commercial nursery, I don't think can tolerate that in their operational process. I mean, maybe they can, they'd have to plan for it. But I mean, obviously, when we match the species correctly, we don't get that. So that would be the ideal. So this is, you know, some of the crossing data from what we've been doing. So this is what data we have to kind of look at what this is. But we think, yeah, we're definitely going to have to pay more attention. And honestly, personally, I was a little surprised that this is so bad, because I kind of thought with the number of hybrids you see all the time, maybe we wouldn't have that much trouble with this. But obviously, with those percentages, you could get a lot of hybrids on the landscape. But again, operational is a different question. That's obviously, I mean, the numbers we're getting out would retain lots on a landscape scale. That percentage retains a lot of hybrids on the landscape. So I think it's kind of like showing both things are true. The hybrids are out there, they're right, but we also do need to worry about them. So we also have a group of families that are not thriving. So they're just, normally it takes about 2 years to get 2 to 3 years to get our trees in our potted nursery big enough to do our bioassays on. We have a group of families that are taking 4 or 5 years. And some are involving admixed parents and some aren't. So we don't know what's going on this. We're going to have to sort this out more. But that's another potential area where we've got at least a question that we're maybe seeing some problems based on this. They just aren't growing. Trees with admixed parents are part of the group that are just not growing well. And, you know, talking about going to outplanting and getting through transplant shock and getting into a competitive situation in a forest, growing half as fast as everybody else is not going to give you a happy ending. So, and again, nursery, there's other potential issues with the nursery. If you want to try to protect a crop in the nursery so that it's not got EB on it before you try to outplant it operationally, you may need to have a species specific protocol for that. We're thinking through when we're trying to find folks who will host a planting, they may have a specific goal for what they want on that property. Because, typically what we're offering is we'll come out, we'll do the planting, we'll manage it during the research phase, and at the end, you get to keep the trees. So they have a goal sometimes of what they want. And if we don't have the species right, we may not be meeting that goal. And then it may take longer for white ash research plantings to really get to the evaluation phase. And so if we promise 12 to 15 years and it takes 20, we may not have a very happy landowner. And we're trying to recruit more people, so we want people to be happy. There's a lot of interest in some of these experiments to doing sampling for other types of experiments. And that adds a layer of risk. If we have a mix of white and green ash in an experiment where we're going to sample for an omics experiment, there's a risk that when you run that analysis, what you're going to find is the species difference. And we may have done this in the past. I'd have to go back through the published papers. The other thing that could happen is because you've got kind of, I wouldn't say apples and oranges, but maybe green apples and red apples, your variance is going to be wider across the whole experiment. And so you risk not finding any significant differences. And so being able to make sure that we can put together these sampling sets in a way that minimizes variance so that we can learn what we're trying to learn is another potential impact. And then just operational things. Our rooted cutting protocol, as an example, works better for green ash than white ash. So if we say, well, I want 25 trees for this planting, we want to generate 25 rooted cuttings. We need to sell a lot more white ash to get to 25 than green. And if we've got the species ID mixed matched, we're not going to get to our goal. And that's just kind of, we have operational pieces of our breeding program in addition to the research experiments. So, this is a little bit of a summary, but as we get ready to look at the orchards, all these problems compound into the next step. When you get ready to say who's going to go into the orchard, we're going to, all these problems that we saw in the experiments are going to come through. And then we have just different groups. We've been talking about these groups. I won't belabor this, but we're focused mostly on green versus white, but we know we've got these other guys out there. And more specifically, if we go to the orange box on this slide, the kind of combinations that our team identified as most trouble for us are green versus white. We all know some are really easy green, some are really easy white, and there's everybody in the middle. So but we want to put populations together that are going to be reproductively competent. So how do we sort these guys out? Green versus pumpkin. It's hard to tell pumpkin in Ohio. Unless you're looking at the seed, which is a mature phenotype, and even then it's a range. Built more in certain not so much the leaves, but the twigs can look kind of green, and built more can be a little tough. And then, we don't even know what we might have as far as some samples from some of these other species. So a tool, I asked the team, what kind of tool do you need? So they said something that can be printed in addition to being available as like a phone guide. They don't want just a phone app. Want something you can print and take to the field. Needs to separate the full species, but also and I don't know, you're probably not going to be separated every tree. I don't know with ash, there's just so much out there. But I think this is what Zane's point was earlier, we need to acknowledge where we have hybrids. So that you're like, okay, well, if I can't tell if this is green and white, well, I know that hybridizes. So maybe it's a hybrid. Maybe it's it's probably good to make notes in the guide. And a lot of taxonomies just describe the pure species. I think this tool should probably talk about hybrids too. That's what the team thought. And not just the summer guide. We use a lot of twig and bud scar characteristics, more or less successfully, you could argue. But when we look at, sometimes we get just seed, and we want to look at the seed. It'd probably be good to have people throw a twig in with seed when they send it in. But it needs to work for different parts of the tree and throughout the year. And I think no one's surprised that our team thinks they need more than one trait to distinguish different species. And this is sort of my comment. You go back into the old papers, you can find examples where I want to say it was the old red ash versus green ash, and it was one has hairs and the other doesn't. And then they grew a provenance study in the nursery, and the hairiness segregated within family. So I mean, herbaria and single samples off the landscape are great, but these things have to be true breeding if they're actually distinguishing species. And as a geneticist, I always think of species as like a breeding population. I don't think of it as just a box to put samples in. I think about who's getting with who. So that's my crossing data, I think, is going to be important. And I think that's one thing we can do is try to help validate potential characteristics by either looking at crosses we've already made or making crosses to look at to make sure things are true breeding. So we would benefit, the sooner we can sort out our population, the better. We've got a few years before I want to rogue an orchard, but I want to stay on track for that. And then, again, I've made the point that it needs to represent populations on the landscape. And I think there's discussion happening about, how do you focus? Do you focus pre-EAB or after? It probably depends where you're at in the country. Obviously in Ohio, we're way past pre-EAB. But yeah, so there's probably, it's going to have to be a conversation and a group decision. So anyway, again, many, many years of work. Partners, I don't even think I have half of the Tree Species in Peril folks on here because I was looking for an orange tea to stick on there and I realized now that it didn't make it. And I think probably some other folks too. So don't feel slighted. It's my ineptitude, not my gratitude for your collaboration that left you off the slide. So yeah, that's the breeding program perspective. So see if there's questions. We have time, or if you just want more coffee.
Yeah, I think it's fine if we cut into the break by a few minutes, especially because we started a little late. So if we want to take a few minutes, if there's questions, that's cool with me.
I have a super quick comment. First, well, a couple comments. At least you remembered to make an acknowledgement slide, whereas I did not. But I mean, we definitely have the same problem where it's such a large group effort. That's the problem to have. The other thing I just wanted to kind of mention is just to sort of plug all of the awesome historical botanical work that's been done. So the papillary trait that we are basically now trying to use to separate the green and white ash species complexes, that's really derived from a long and storied history of botanists taking close looks at this material. And then me just having a big pile of sequencing data, understanding that I can't tell them apart, and going back to the primary literature and just saying, I wonder if I can correlate these things. And then sure enough, comes out pretty nice and clean. So that's where that originates. I just wanted to make it clear that I can't take any.
You gotta love Jonathan, right? He's been dead for years, but you gotta.
Yeah. Well, folks like Alan Weekly have done some
Yeah.
phenomenal work.
Among others.
Yes, absolutely.
Online question from Natalie, I think.
Yeah, Natalie, if you want to go ahead and unmute yourself.
Yes, hello, thank you. Thank you, Mary. I put my my question in the chat, but I don't know if they were actually, if you were able to see them. Thank you very much for your very informative presentation. I think you have progressed a lot. That's amazing. From time to time, what I see, it's actually things are moving forward. Different question for you. In terms of crossing compatibility, have you tested the polymix in terms of control crossing? Usually it can increase the germination rate, having like a number of genotype competing for the for the fertilization. So have you tested that?
We haven't done much polymix. So what she's asking about is mixing several pollens together to do the control cross. So
Yep.
we're at this stage, we're really focused on doing control cross families in order to do our heritability estimates.
Mm-hmm.
But I think the polymix would be a better approximation of obviously the seed orchard productivity. I think what we'll probably do instead of a lot of polymix work is rogue or thin out that Holden orchard and then maybe do open pollinated seed from each of the mothers.
Okay.
To get an estimate of what we're to see. And that would, we're probably close enough to that. We can probably get that just a couple years later than if we started polymix now.
Okay.
So that's
Okay.
my thought on that initially.
Okay. And in terms of flowering phenology, have you seen a lot of variation, like within each species and between species? Is there a window of opportunity that vary a lot, or?
The phenology, yeah, I mean, they're a little different, but they overlap so much
Okay,
between the
okay,
different
okay.
species. The green and white are very overlapping. So I don't think that at any particular locality, the phenology is probably not separating the species.
Okay, and perhaps one last, I would like to know if you envision a genomic selection in the future, considering that you will have all the parent sequence and
Yeah, I think we'll do as much as we can get Meg to do for us. No, we have some, I mean, I think it's the seriousness. I mean, we've been having some conversations because there's a lot of, like I said, I think it's a tremendous opportunity to have all this deep genomic sequencing on our entire breeding population. I didn't expect to have that. So I think what we can do with it, I think it's just getting done. We're sorting out the IDs first. That's been like the panic moment. Oh my God, we got to get these trees sorted out. I think then we can calm down and be like, what else can we do with this giant pile of data and this big like bunch of trees we have planted? And I think there's going to be a lot of opportunities. Just for example, another thing we were talking about, a lot of our green ash are from an area that Kathleen Oak Openings, Swan Creek, that Kathleen did a huge monitoring project in. We identified a whole group of lingering ash. I've always wondered if those were related to each other. With whole genome sequencing, we can know.
yeah.
So, you know, that's just an example of something that's like, oh, it'd be nice if I knew. And it's like, oh, wait a minute, I can actually know. So I think, yeah, maybe we should chat about how you think genomic selection, you know, we would love to hear your ideas on how to leverage this data.
Okay.
For the
Well,
group
I
that
hope
doesn't
to.
know, she's one of the people whose programs, I looked at the heritabilities and what she works in those guys. She's ahead of us and very successful. So
No, that's okay. But just I hope that I'll be there in May. So I don't know if you're gonna be at the Trees in Peril meeting in May. So anyway, we can have a chat
awesome.
and so congratulations for your work.
Thank you. Yeah.
Again, I think I'm making a good here, because the overall objective of doing all this work is to select a resilient specimen that you will get breed from.
I would like 40 to 50 individuals in an orchard producing very diverse seed to send to a nursery to use in a region to restore forests.
Region.
Yeah, so I mean, and then we'll have to do experiments to see how far you can take that seed. There's going to be a limit. So yeah, that not one, I want a pile. So yeah. And again, that's to make sure we have retained all of that genetic diversity that I started with for local adaptation and every other pest that we were just hearing about at the diagnosis center, because all that stuff's out there still too. And trust me, when you grow a whole bunch of ash, you find all those guys. Thank you.
All right, thank you. Let's see, now we are scheduled for a break. What time is it right now? 3:40.
I'm sorry, 3:20.
Okay, 3:20.
I added 5 to the wrong.
Okay. Do we want to take a 15-minute break and start 5 minutes late, or are people cool with a 10-minute break? How are you all feeling here in person? 10 is fine. Okay, cool. So we'll break now and we will be back at 3:30.
Oh.